We have part 2 now of pregnancy and human development, page 1066. And we just had that moment of fertilization, and then, with special regard to capacitance, the idea that in order for fertilization to take place, that the enzymes at the forward tip end of the sperm has to become available to break through the outer surrounding corona radiata, consisting of granulosa cells and zona pellucida, in order for there to be a sperm which then actually performs the fertilization. So in this case, let me just sort of briefly come over here to this page and present the idea that we have all of these sperm that arrive. It's going to take at least 50 sperm to be nearby and depositing their acrosome. So the early bird does not necessarily catch the worm, so to speak, or that is to say, it could be the 50th caller that actually is the one that makes it to the fertilization event. So when finally there is an opening created by the many acrosomes that have been needed to do the enzymatic digestion, then finally one sperm will actually be able to make contact with the membrane. Ultimately, over here we can see that the contact has been made, and that over here there's going to be that moment of fertilization. It's a little bit like infection, if you think about it from that perspective. So the nucleus here from the sperm has to be released ultimately into the cytoplasm of the ovum. Notice that what's being cut off here, there's a little bit of mitochondria from the sperm sometimes that might be included in this new zygote, but for the most part, the zygote contains everything that's needed for actually many cell divisions. So there's many cell divisions. Maybe there's 50, 100, who knows how many. Well, in terms of the blastocyst, you kind of get the idea that there's hundreds of cells worth of organelles and cytoplasm available specifically from the ovum, which means that a great deal of the heritage of any individual comes from mom, right? So the mitochondria mostly are from mom, the cytoplasm is from mom, and only the 23 chromosomes delivered by the sperm is from dad, right? So the vast majority, mitochondrial DNA, for the most part, is mom. There could be this rare moment of a little bit of introduction of a mitochondria, but for the most part, this is entirely, all the organelles belong to the mother and the mother's heritage and can be traced through the maternal line. They do talk here about then the need for monospermy. This is a dosage regulation issue in which only one sperm is permitted to enter the egg, because only 23 chromosomes then can combine with 23 chromosomes from the mother to create the 46 chromosome individual of the zygote. And so there are these blocks that prevent polyspermy from occurring. Okay, and we have this thing called the cortical reaction. And that's a very quick and sudden event. There are more elaborate events that take place after that that involve some, how would I say, sealing off of that area where the entry took place. But there was a really quick calcium activated. So it says a calcium surge, I can't get rid of there we are, also causes the cortical reaction, which is that rapid enclosure kind of thing. Okay, so that's the deal, and I want to emphasize that as you can see, the difference between the sperm and the egg itself is quite vast. This is literally a terrain of what is to become many dozens of cells, daughter cells, from the original zygote. And the organelles in the cytoplasm are being provided by the oocyte. This outer layer here, called the corona radiata, is made up of granulosa cells and then zona pellucida. So that's something that has to be penetrated, and then it seals up again. So ultimately, even after we have the fertilization event, which is depicted here again, there's going to be a period of time where the ovum is going to divide and divide and divide, and it has not yet hatched until it gets a little ways along. So we'll take a look at where that is. But here in this moment, we get to see that this is referred to as the pronucleus that's delivered by the sperm. Here we have the female pronucleus. She's just had yet another, as the fertilization event was going on, she is just having yet another cell division. Here is a little lineup right here, a little metaphase moment. And then finally we get this next polar body, the last polar body. There should be 3 of them, and then this is the pronucleus. So 3 polar bodies plus the ovum is the result. Then they get ready for their first mitotic division, and this sort of unifies everything. We have the prophase moment here, and then ultimately we have the first division. And we come over here and we take a look, and we see here is the oocyte deposited here after ovulation. Here's the moment of fertilization. Here is that zygote, which is the fertilized egg. So the egg with 23 chromosomes, with the additional sperms 23, becomes the 46 chromosome zygote. And then this is the stable moment here. That first division, which divides one cell into 2 cells, could result in identical twinning. That is to say, the 2 cells could actually separate, and there could be, in fact, identical twins as a result of that, 2 completely separate individuals. So we don't actually have a stable program until we get to the 4-cell stage. So it's the second mitotic division that leads to 4. And now this is the stable phase. For those of you that have studied microbiology or molecular biology of human development, there is a whole phylogenetic history of species that we all have to get to the 4-cell stage. And many things can be predicted by those 4 cells. Only one of them has acetylcholine, which is destined to become musculature and mesoderm. So there's a lot of interesting things going on here. So this is the stable moment, and I've made that notation here. And then we get multiple divisions further after that. And notice that we're still in progress now. This is a ciliated epithelial tissue lining here that is gently brushing the developing blastocyst, in this case a morula, we call it. So we have the morula, which is the 3-day unit. We don't actually get to the next stage here, which is as you can see, a blastocyst. We have a little cavity in there. So we have here it's solid cells all the way through. Here it's just this region that has a solid area of cellular development, and then the very outside is referred to as a trophoblast. And so we're just about to do the implantation, and I'll pick that up in our next section here.